Welcome everyone. Today we'll be covering the reproductive system. This system is unique in that it is not essential for individual survival, but it absolutely vital to the continuation of the species. We'll explore how the structures and function of both the male and female reproductive systems, how they differ, how they coordinate, and how the hormones play a role in regulating everyone from puberty to reproductive aging. By the end of this lecture, you should have a strong understanding of how the anatomy supports key physiological processes like gamete production, fertilization, and pregnancy. Let's start by taking a broad look at the purpose and components of this complex and fascinating system. Before we dive into the content, let's review the key learning objectives for this chapter. First, you should be able to describe the detailed anatomy of both the male and female reproductive systems, including the ducts, glands, and external structures that support production. We'll also focus on the hormonal regulation of these systems, especially how the hypothalamus and the pituitary glands orchestrate reproductive function through FSH, LH, and other hormones. You'll be expected to trace the journey of sperm cells from its origin in the testes all the way to the potential fertilization in the uterine tubes. We'll also cover the cyclical events in the ovaries that lead up to ovulation, including follicle development. Finally, we'll discuss the major physical and hormonal changes that occur during puberty, how the reproductive organs mature, and how secondary sex characteristics begin to appear. Keep these objectives in mind as we move through the material. The reproductive system is unique in that it is not essential for individual survival, but it is crucial for the survival of the species. Its primary role is to produce, nourish, and transport gametes, which include the sperm in males and the ova in females, and ultimately support the union of these cells to form a new life. At the center of the system is the gonads, the testes and the ovaries, which not only generate gametes but also secrete the sex hormones that drive puberty, secondary sex characteristics, and reproductive cycles. The secondary structures include ducts, glands, and external genitalia, work together to ensure these processes occur efficiently. Hormones tightly regulate each stage, making this system a perfect example of physiological integration. The male reproductive system is designed to produce, mature, and deliver sperm. The testes, housed in the scrotum, are the primary organ where sperm and testosterone are produced. From there, sperm travel to the epididymis, where they gain the ability to swim and fertilize an egg. During ejaculation, sperm passes through the vas deferens, merge with secretions from the seminal glands and at the ejaculatory duct, and enter the urethra. Along the way, the accessory glands contribute fluid that nourishes and protects the sperm, forming semen. The penis, which contains the urethra, serves as the delivery system for this reproductive fluid, making the coordination of these structures essential for male fertility. The scrotum is a skin-covered sac that supports the testes and plays a key role in temperature regulation. It lies outside the body cavity, allowing the testes to remain about 2 to 3 degrees Celsius cooler than the body's core, which is an essential condition for effective spermatogenesis. The scrotum is divided internally into 2 compartments, each containing 1 testis. Externally, this division is marked by a scrotal raphe, which is a visible ridge running along the midline. Thermal regulation is managed by 2 muscles, the dartos, which causes the skin to wrinkle and reduce surface area, and the cremaster, which raises or lowers the testes in response to temperature changes. This system ensures a stable environment for sperm development. Within the testes, a thick fibrous capsule called the tunica albuginea surrounds and protects the organ. It projects inward to divide the testis into roughly 250 lobules. Inside each lobule, there's 1 to 4 seminiferous tubules where sperm development takes place. These tubules are tightly coiled to maximize surface area for efficient sperm production. In between the tubules are clusters of interstitial cells, also known as Leydig cells. These cells respond to luteinizing hormone by producing testosterone, which is the key hormone responsible for male secondary sex characteristics, libido, and continued spermatogenesis. The organization of these structures ensures both hormonal and reproductive function occurs in close coordination. Spermatogenesis is the multi-step process of developing mature sperm, and it all starts with spermatogonia, which are diploid stem cells found near the basement membrane of the seminiferous tubules. These divide mitotically to maintain their population and produce primary spermatocytes, which enter meiosis. In the first meiotic division, they become secondary spermatocytes, and after meiosis II, they give rise to haploid spermatids. Spermatids then undergo structural transformation called spermiogenesis, where they develop their tails, acrosomes, and streamlined bodies to become functional spermatozoa. Although the entire process takes roughly 64 to 72 days, it's continuous, ensuring a constant supply of new sperm. Alongside the germ cells, the seminiferous tubules contain Sertoli cells. These are tall columnar cells that span the width of the tubule wall. These are essential for supporting, nourishing, and protecting developing sperm. They also form the blood-testis barrier, which prevents the immune system from attacking the genetically unique sperm cells. Sertoli cells are stimulated by FSH and secrete inhibin to help regulate feedback in the hypothalamic pituitary gonadal axis. Meanwhile, LH stimulates Leydig cells to release testosterone, which supports the entire process of spermatogenesis. Together, these cellular and hormonal interactions ensure that sperm development is both efficient and tightly regulated. The mature sperm, or spermatozoon, is highly specialized for delivering genetic material to the egg. The head contains the condensed nucleus with 23 chromosomes, representing the male's genetic contribution. Covering the head is the acrosome, which is a modified lysosome filled with enzymes that help the sperm penetrate the outer layer of the oocyte during fertilization. The midpiece is loaded with mitochondria, which produce ATP necessary to power the tail, which is flagella that enables the sperm to swim. This unique design gives sperm the motility and precision needed to navigate the female reproductive tract and reach the egg. Sperm are produced in the seminiferous tubules of the testes and then pass into the epididymis, where they mature and become motile. During ejaculation, sperm travels through the vas deferens, enter the ejaculatory duct, pass through the urethra, and are expelled via the penis. Once inside the female reproductive tract, they can survive for up to 5 days, though fertilization usually occurs within the first 24 to 48 hours. Before a sperm can fertilize an egg, it must undergo capacitation, which is the final maturing step that ensures and occurs within the female tract. This process enhances motility and prepares the acrosome to release its enzyme, enabling the sperm to penetrate the oocyte's protective layer. After leaving the seminiferous tubules, immature sperm enter the epididymis, which is a long coiled duct that lies snugly against the back of the testis. Although it appears compact, it's nearly 6 meters long when fully uncoiled, giving sperm ample time to mature. This is where sperm acquire motility and the capacity to fertilize an egg, two key changes that don't occur during spermatogenesis itself. The epididymis also acts as a storage site, holding sperm for several weeks until ejaculation. If not used, these sperm are broken down and reabsorbed in the body. The structure plays a critical role for maintaining health and functional sperm population. When ejaculation is triggered, sperm are propelled from the epididymis into the vas deferens, which is a firm muscular tube that travels within the spermatocord and enters the pelvic cavity through the inguinal canal. As it nears the prostate, the vas deferens expands into the ampulla, where some sperm may be briefly stored, and then it joins the ducts of the seminal vesicle to form the ejaculatory duct. This short duct pierces the prostate and empties into the prostatic urethra. During ejaculation, rhythmic muscular contractions push sperm and glandular secretions into the urethra, preparing the mixture for final delivery. These connections highlight the precision and timing needed for successful semen transport. The majority of semen volume comes from accessory glands, not the sperm themselves. The seminal vesicles are a pair of glands located just behind the bladder, and they contribute about 60% of semen. Their fluid is alkaline to neutralize vaginal acidity and contain fructose to energize sperm. Prostaglandins support sperm motility and may help stimulate uterine contractions that draw sperm inwards towards the egg. Clotting proteins form a temporary coagulum that helps semen stay in the reproductive tract after ejaculation. The prostate gland, which surrounds the urethra just below the bladder, adds a milky secretion rich in enzymes like PSA that liquefies semen after it clots. Zinc from the prostate plays a protective role, stabilizing and preserving sperm DNA. The last accessory glands are the bulbourethral gland, also known as the Cowper's gland. These small glands sit beneath the prostate and secrete a clear alkaline mucus during sexual arousal. This pre-ejaculate neutralizes any acidic residue in the urethra from urine and lubricates the passage for the semen that follows. Altogether, semen is a complex mixture of fluids designed to support and protect sperm on their journey. Its slightly alkaline pH helps counteract the acidic environment of the vagina, and the mixture includes sugars, enzymes, buffers, and antimicrobial substances. A typical ejaculation releases 2 to 5 millimeters of semen containing tens of millions of sperm, through only one is needed for fertilization. The penis functions as both the organ of sexual intercourse and the terminal portion of the urinary tract. Structurally, it's composed of 3 columns of erectile tissue. 2 corpus cavernosa run along the dorsal side, and a single corpus spongiosum runs ventrally and supports the urethra, preventing it from closing during erection. The distal tip of the penis forms a glans, which is rich in sensory receptors and often covered by a fold of skin called the prepuce or foreskin in uncircumcised males. During arousal, vascular spaces in the erectile tissue fill with blood, causing the penis to become rigid, allowing it to deliver semen effectively into the female reproductive tract. Erection begins within the parasympathetic stimulation that causes arteries in the erectile tissue to dilate, allowing blood to fill the vascular spaces. At the same time, veins are compressed, trapping blood and maintaining the erection. Ejaculation, on the other hand, is driven by the sympathetic reflex. It occurs in 2 phases: emission, that is when sperm and glandular fluids move into the urethra, and expulsion, when the rhythmic contractions of pelvic muscles propel semen out of the body. This entire process involves precise coordination between the nervous and circulatory systems, allowing for successful delivery of sperm during sexual activity. Male reproductive function is tightly controlled by the hypothalamic pituitary gonadal axis. It begins in the hypothalamus, which secretes GnRH in a pulsatile rhythm. This hormone stimulates the anterior pituitary to release 2 gonadotrophins, LH and FSH. LH acts on the interstitial Leydig cells in the testes, prompting them to produce testosterone. FSH works on the Sertoli cells within the seminiferous tubules, supporting the process of spermatogenesis. Together, these 2 hormones ensure both the structural and functional aspects of sperm development are regulated. This system is highly responsive to feedback, maintaining hormonal balance throughout adulthood. Testosterone plays a central role in male physiology. Beyond maintaining spermatogenesis, it drives the development of secondary sex characteristics like facial hair, deeper voice, and increased muscle mass. It also supports libido and bone density. As testosterone levels rises, they exert negative feedback on the hypothalamus and pituitary, suppressing further release of GnRH and LH. Additionally, Sertoli cells release inhibin when sperm production is deficient, which selectively inhibits FSH secretions. This dual feedback mechanism ensures that hormonal levels remain within a narrow range, preserving fertility while preventing overproduction. It is a highly efficient loop that balances reproductive demand with physiological health. The female reproductive system is designed for oocyte production, fertilization, and support of embryonic and fetal development. The ovaries, which are the primary sex organs, are responsible for producing and releasing oocytes in a monthly cycle, as well as secreting the hormones estrogen and progesterone. Once released, the oocyte enters the uterine tube or fallopian tubes, where fertilization typically occurs. The oocyte or fertilized egg then travels to the uterus, which is the muscular pear-shaped organ where implantation and development take place. If pregnancy does not occur, the uterine lining is shed during menstruation. The vagina serves multiple roles. It receives the penis during intercourse, allowing menstrual fluid to exit, and functions as the birth canal. Externally, the vulva includes structures such as the labia and the clitoris, which play roles in protection and sexual arousal. Finally, the mammary glands, while not directly part of the internal reproductive tract, are vital postnatally for nourishing the newborn through milk production. Together, these organs coordinate reproductive, hormonal, and maternal functions across the lifespan. The vagina is a highly adaptable muscular tube that connects the cervix of the uterus to the external genitalia. It serves multiple essential roles, is the receptacle for the penis during intercourse, provides the exit pathway for menstrual blood, and functions as a birth canal during labor. Structurally, it has 3 layers. The mucosa is lined with stratified squamous epithelium that protects against friction and is arranged in folds, or rugae, to allow expansion. The muscularis gives the vagina its strength and flexibility, while the adventitia anchor it to the surrounding tissues. The vaginal environment is normally acidic due to lactic acid produced by resident bacteria, which is an important defense against infections. The ovaries are the primary reproductive organs in females, responsible for producing both oocytes and sex hormones. Each ovary is held in place by a set of ligaments that anchor it to the uterus and pelvic wall. The surface is covered by a thin layer of germinal epithelium and a dense tunica albuginea beneath. Internally, the ovaries is divided into a cortex and medulla. The cortex is where oocytes containing follicles are located at various stages of development. The medulla contains blood vessels and lymphatics and nerves that support ovarian function. Hormones secreted by the ovaries, mainly estrogen and progesterone, are essential for regulating the menstrual cycle and preparing the uterus for potential pregnancy. Oogenesis is a process of which the female body produces the mature egg cell. It starts before birth, when oogonia divide mitotically and become primary oocytes. These oocytes begin meiosis 1, but halt at prophase 1 and remain in this suspended state until puberty. At birth, a female has around 1 to 2 million primary oocytes, but at puberty only 400,000 remain. From then on, roughly 1 oocyte per month resumes meiosis in response to hormonal cues. Unlike spermatogenesis, which continues throughout a man's life, oogenesis is cyclical and limited, with only about 400 oocytes ever completing ovulation. Each month, a few follicles begin to mature, but typically only one reaches full development. The follicle progresses through several stages, starting at the primordial follicle and develop into a primary, then secondary, and finally into a vascular or Graafian follicle. During ovulation, which is triggered by a surge of LH, the mature follicle ruptures and releases a secondary oocyte into the nearby uterine tube. The empty follicle transforms into the corpus luteum, which acts as a temporary endocrine gland by secreting progesterone to maintain the uterine lining. If fertilization does not occur, the corpus luteum degenerates into a fibrous structure called the corpus albicans. The hormone level declines, leading to menstruation. Follicular development is a stepwise process that prepares a single oocyte for ovulation during each menstrual cycle. Females are born with all of their primordial follicles, which are simple structures consisting of primary oocytes surrounded by a single layer of flat cells. Once activated, a follicle becomes primary, where granulosa cells become cuboidal, and then the zone of pellucida begins to form around the oocyte. As the follicle grows, it enters a secondary stage, developing more layers of granulosa cells and acquiring the theca cells that will develop and produce estrogen. Fluid pockets begin to form between the granulosa cells, eventually coalescing into a large antrum. This marks the vesicular or Graafian stage, where the follicle bulges at the ovarian surface and is ready for ovulation. After ovulation, the ruptured Graafian follicle doesn't simply collapse. It transforms into the temporary endocrine structure called the corpus luteum. This structure plays a critical role for the secondary half of the menstrual cycle by secreting progesterone and some estrogen to maintain the thickened endometrial lining. If fertilization and implantation occur, the embryo secretes HCG, human chorionic gonadotropin hormone, which preserves the corpus luteum and keeps hormone levels high. But if pregnancy does not occur, the corpus luteum degenerates after about 10 to 14 days and becomes the corpus albicans, which is a small white fibrous remnant. As hormone levels decline, the endometrial lining is shed, marking the beginning of a new cycle. The follicular phase is the first half of the ovarian cycle and begins on the first day of menstruation. During this time, FSH from the anterior pituitary stimulates multiple follicles in the ovary to begin maturing. Usually only one of these becomes the dominant Graafian follicle. As it develops, granulosa cells around the oocyte secrete increasing amounts of estrogen, which stimulate regeneration of the endometrial lining. Estrogen levels eventually reach the threshold that trigger a surge of LH, leading to ovulation, the release of the secondary oocyte around day 14. This phase prepares the egg and the uterine lining for a possible pregnancy. The luteal phase begins immediately after ovulation and spans the second half of the ovarian cycle. The ruptured follicles transforms into the corpus luteum, which secretes progesterone and a smaller amount of estrogen. Progesterone is essential during this phase. It stabilizes and thickens the endometrium, creating a hospitable environment for implantation. If fertilization does not occur, the corpus luteum degenerates into the corpus albicans. Hormone levels fall, and the cycle restarts with menstruation. However, if implantation does occur, the developing embryo releases HCG, which preserves the corpus luteum and supports early pregnancy. The uterine tubes are muscular, ciliated passageways that connect each ovary to the uterus. They don't physically touch the ovary, but the funnel-shaped infundibulum with its finger-like fimbriae help guide the ovulated oocyte into the tube. The ampulla is the widest and longest segment and usually where fertilization takes place. As the oocyte travels through the tube, ciliated epithelial cells in the lining help sweep it towards the uterus. At the same time, gentle peristaltic contractions of the smooth muscle assist in the movement. These, combined with mechanisms, are essential for transporting both the egg and, if present, the sperm. These fimbriae are these delicate finger-like extensions located at the distal ends of the uterine tube, specifically on the infundibulum, which is the funnel-shaped region nearest the ovary. These structures don't physically connect to the ovary, but play a critical role during ovulation. When a mature follicle ruptures and releases an oocyte, the fimbriae move rhythmically, guided by the cilia and the smooth muscle contractions to sweep the oocyte into the uterine tube. This is an essential step in reproduction, as it allows the oocyte to enter the tube where it may encounter sperm and undergo fertilization. Without effective fimbriae action, the oocyte might remain in the peritoneal cavity and never reach the uterus. Fertilization usually takes place in the ampulla within 24 hours after ovulation. Sperm must reach the oocyte during this brief window, and once one successfully penetrates, the oocyte completes meiosis II, forming a zygote. The zygote then begins a series of rapid mitotic divisions, which is the process called cleavage, as it continues its journey towards the uterus. This trip takes several days, which the early embryo transforms into the blastocyst. When it reaches the uterus, typically around day 5 or 6, it begins the process of implantation into the uterine lining. This marks the start of pregnancy, assuming all conditions are favorable. The uterus is a central organ for a female reproductive system, designed to support implantation, pregnancy, and childbirth. It sits in between the bladder and the rectum and is divided into distinct regions: the fundus, which is at the top, the body, and then the narrowing isthmus, and the cervix, which projects into the vagina. Structurally, it has 3 layers. The perimetrium is a thin outer covering, while the myometrium makes up the bulk of the uterine wall, with smooth muscle that contracts during labor. The innermost layer, the endometrium, is where the fertilized egg implants. It consists of 2 layers: the stratum functionalis, which thickens and is shed each month, and the stratum basalis, which remains behind and regenerates the lining after each cycle. The menstrual cycle prepares the female body each month for the possibility of pregnancy. The cycle begins with menstruation, the visible shedding of the uterine lining that occurs when no fertilized egg implants. This is considered day 1 of the cycle. During the proliferative phase, estrogen from developing follicles stimulates the endometrium to regrow. Ovulation occurs around day 14. Afterward, the secretory phase begins with the progesterone from the corpus luteum maintaining a thick, nutrient-rich lining. If no fertilization occurs, hormone levels drop, the corpus luteum regresses, and the functional layer of the endometrium breaks down and is expelled as the next period begins. The menstrual cycle is divided into 3 coordinated phases that prepare the uterine lining for possible implantation. It begins in the menstrual phase, which typically lasts 5 days. Here, the functional layer of the endometrium is shed due to the sharp drop in progesterone and estrogen. Once menstruation ends, the proliferative phase begins. During this stage, estrogen secreted by the developing ovarian follicle stimulates regrowth of the stratum functionalis. Endometrial glands lengthen, spiral arteries regenerate, and the linings thicken to prepare for a potential pregnancy. This phase concludes with ovulation, when a mature oocyte is released from the ovary. Ovulation is the midpoint event of the menstrual cycle and marks the release of the secondary oocyte from the ovary. It is triggered by a dramatic surge in luteinizing hormone, usually around day 14 of the typical 28-day cycle. This surge causes the mature Graafian follicle to rupture, releasing the oocyte into the peritoneal cavity near the uterine tubes. The fimbriae of the uterine tubes gently sweep the oocyte into the tube where it begins this journey towards the uterus. This oocyte remains viable for fertilization for about 24 hours, making the timing of ovulation critical for conception. The remnants of the follicle then form the corpus luteum, which supports the uterine lining in the second half of the cycle. After ovulation, the secretory phase begins. The ruptured follicle transforms into the corpus luteum, which secretes large amounts of progesterone and some estrogen. These hormones cause the endometrium to reach peak thickness, with coiled glands secreting glycogen-rich fluid and spiral arteries becoming more prominent. This environment is ideal for implantation if fertilization occurs. If not, the corpus luteum degenerates, hormone levels drop, and the functional layer is no longer maintained, leading to the start of the next menstrual cycle. This entire process is controlled by the interplay between the pituitary hormones FSH and LH and the ovarian hormones estrogen and progesterone. The vulva includes several external structures that offer both protection and sensory function. The mons pubis is a rounded fatty area overlying the pubic bone that provides cushioning during sexual activity. Just beneath it are the labia majora, which are prominent skin folds that serve as outer protective barriers. These folds are typically covered in pubic hair and contain oil and sweat glands to help maintain skin health. Inside the labia majora are the labia minora, which are more delicate, hairless folds rich in sensory innervation and blood supply. They help form the boundaries of the vestibule and contribute to sexual sensitivity while shielding the urethral and vaginal openings. At the upper margin of the vestibule lies the clitoris, which is a highly sensitive structure composed of erectile tissue and packed with sensory receptors. Like the penis, it becomes engorged during arousal and is partially covered by a fold of skin called the prepuce. The vestibule itself is the central space between the labia minora and houses both the external urethral orifice and the vaginal opening. On the other side of the vaginal openings are the greater vestibular gland, also known as the Bartholin's glands. These produce mucus during sexual arousal to lubricate the vulva and vaginal openings, facilitating comfortable intercourse. These components of the vulva are critical not only for protection but also for sexual function and reproductive readiness. Mammary glands are specialized exocrine glands located within the breast tissue, lying over the pectoralis major muscle and embedded in fat. Structurally, each breast is divided into 15 to 20 lobules, and with each lobe there are smaller lobules that contain milk-secreting alveoli. These alveoli are lined with epithelial cells that respond to hormonal signals by producing milk. Milk then travels through the branching network of lactiferous ducts, which converge into a larger duct that expands slightly into lactiferous sinuses before opening in the nipple. Surrounding the nipple is the areola, which is a pigmented region that contains sebaceous glands to lubricate and protect the skin during nursing. Milk production and release are tightly regulated by hormonal signals. Prolactin, secreted by the anterior pituitary, stimulates the alveolar cells to synthesize and secrete milk. After birth, suckling from the infant triggers the release of oxytocin from the posterior pituitary. Oxytocin causes the myoepithelial cells surrounding the alveoli to contract, pushing milk through the ducts towards the nipple. This is known as the let-down reflex. These hormonal effects are part of a positive feedback loop. The more frequently the infant nurses, the more prolactin and oxytocin is released, maintaining a steady supply of milk. This system ensures that milk production matches the infant's nutritional needs. Female reproductive hormones are coordinated through the hypothalamic pituitary ovarian axis. The hypothalamus secretes GnRH impulses, which stimulates the anterior pituitary to release FSH and LH. FSH promotes the growth and maturation of ovarian follicles, while LH plays a key role in triggering ovulation. Then, the release of the secondary oocyte from the dominant follicle. After ovulation, LH also supports the transformation of the ruptured follicle in the corpus luteum, which is vital for secreting hormones released to maintain a uterine lining. These hormonal surges and declines are closely regulated by feedback mechanisms involved estrogen and progesterone. Estrogen and progesterone are key ovarian hormones that regulate the menstrual cycle. Estrogen levels rise during the follicular phase, promote the thickening of the endometrial lining, and contribute to female secondary sex characteristics like breast development and fat distribution. Interestingly, moderate estrogen suppresses FSH and LH, but a sustained high levels just before ovulation reverses this and causes LH surge. After ovulation, progesterone becomes the dominant hormone. It prepares and maintains the endometrium for potential implantation and strongly inhibits further GnRH, FSH, and LH release to prevent another ovulation during the same cycle. Inhibins secreted by the granulosa cells help fine-tune this balance by specifically inhibiting FSH. Puberty marks the transition from childhood into reproductive maturity. It begins when the hypothalamus increases GnRH secretion, stimulating the pituitary to release FSH and LH. In females, these hormones activate the ovaries, leading to breast development, growth of body hair, and eventually menarche, the first menstrual period. Over time, ovulation becomes regular. In males, LH and FSH stimulates the testes, causing growth of the penis and testicles, deeper voice, facial hair, and beginning of sperm production. These changes also involve increased secretions of sex hormones, estrogen in females and testosterone in males, that drives physical and emotional maturation. As individuals age, the reproductive system undergoes significant changes. In females, menopause marks the end of the reproductive capacity, usually occurring between the ages of 45 and 55. Ovarian follicles are depleted and the ovaries stop responding effectively to FSH and LH, leading to decreased estrogen and progesterone. This hormonal shift can cause hot flashes, deep sleep disturbances, mood swings, and increased risk of osteoporosis. In males, reproductive aging is more gradual and sometimes called andropause. Testosterone levels slowly declines, which can affect libido, energy levels, and muscle mass. However, many men retain the ability to produce sperm well into their older years. The reproductive system in males and females are structurally distinct, but functionally complementary. In males, the testes are the primary organs producing both sperm and testosterone. A system of ducts and glands ensures sperm are matured, nourished, and delivered through the penis. In females, the ovaries produce oocytes and hormones that regulate the cycles and prepare the uterus for possible implantation. Fertilization usually occurs in the uterine tubes, with the uterus providing support for the developing fetus. The vagina serves as the entryway for sperm and the exit for both menstruation and childbirth. Externally, structures like the labia and the clitoris contribute to protection and arousal. After childbirth, the mammary glands become vital for newborn nutrition through milk production. Together, these systems ensure the reproductive success and hormonal balance through the lifespan.